Okay, welcome everyone. Thanks for letting us allow some folks to trickle in. We're glad to have all of you joining us today for the launch of this new series. We're launching this series, the weekly Lunch and Learn series, to give folks a chance to kind of learn more about our work directly from the scientists who are on the forefront of the research every day and really turning the wheels and pushing the work forward. So we're honored to have week one here with Dr. Kathryn Turner. She's going to be leading us off. So thanks for being a part of this inaugural group. Before we get started, we wanted to cover some just kind of basic details on the layout of the webinar today. So you probably noticed by now we'll be muting all participants during the presentation, but we welcome and encourage all participation and comments. We're going to have some time for questions. During and after the presentation, please feel free to direct message me. My name's Will Schneider. Utilizing the chat function, you can send me a message and I'll be sure to make sure Kathryn gets the question. And afterwards, we'll be facilitating some Q&A. So we'll have a little bit of limited time to answer questions, but we're going to do our best to get to as many as possible. We just ask that you keep your questions focused on the work presented and using Kathryn's research and taking away things from her talk today to kind of learn more about our Silphium program and the crop protection genetics program. So just one final note, this presentation is being recorded, so just wanted to let you all know there. But other than that, we'll get started. Today's presenter is Dr. Kathryn Turner. Kathryn leads our crop protection genetics program. She's originally from Oklahoma, but was first introduced to the Land Institute as a summer intern while attending Austin College, where she studied biology and environmental studies. During her TLI internship in 2005, she developed an interest in plant breeding, and after graduating from Austin College, went to pursue her graduate studies at the University of Minnesota, where she focused on identifying disease resistance in both annual wheat and perennial wheat. Thereafter, she was hired as a postdoctoral fellow under Shuwen Wang, one of our lead scientists, to better understand the genetics of perennial wheat. And while working on perennial wheat, she also researched disease problems across a multitude of the crops we're developing. This research led her to her current position, where she focuses solely on crop protection genetics. And in 2018, she became our first research associate, heading a new program to understand, identify, and control the diseases in our crops. So without further ado, I'm going to turn it over to Kathryn.
Okay, I think I'm unmuted now. I think we got that figured out. Good. Well, thanks for everyone for joining us today. I thought it would be kind of a nice break to talk about a different kind of virus than the one that's all over the headlines right now. And I figured there are going to be a few parallels, but mostly we'll talk about plant viruses, and in particular, a virus that I'm working on in Silphium, which is related to sunflower and pictured on the slide. So I'll give just a little bit of overview about viruses, and then kind of jump into the work that we're doing right now on this particular virus of Silphium. So viruses are kind of interesting. Like other living organisms, they have DNA and RNA, but they are not quite the same as other living organisms in that they don't have cells, and they also only can replicate when they're within other living hosts, and they're essentially non-living or inert outside of their living host. And viruses are the most abundant biological entity on Earth. In the ocean, in particular, they have their viral particles are orders of magnitude greater than any other life forms. And a lot of those viruses are actually viruses of bacteria and other microbes. In the human genome, viruses make up about 8% of all of the DNA, and it's mostly in the form of fragmented viral DNA, so it's not causing disease. It's just kind of left over. We often think of viruses as agents of disease, but not all cause disease. Some of them have very little effect, and some of them may actually be beneficial. In plants, they can help with nutrient uptake, and can enhance drought tolerance. Um, for There's been a study done in mice where mice infected with viruses can actually uh, be resistant to bacteria that cause the bubonic plague. So we're kind of learning more about microorganisms, like the beneficial gut microbes in our intestines, and mycorrhizae, fungi, and plants that help with nutrient uptake. So a lot of the organisms that we typically associate as causing diseases may have some other benefits. But for most of this presentation, I'm going to talk about viruses as pathogens. And they're responsible for about 50% of emerging plant diseases, and they're detected pretty often when sampled just in plants that don't necessarily have symptoms. So about a quarter to a third of plants in grasslands, marshes, forests across all biomes have viruses that are infecting them but may not show symptoms. And as we bring those crops or plants into cultivation, like as we're domesticating new species, the abundance of viruses tends to go up in cultivated areas. So we can expect, as we bring our domesticated plants into field production, we may start to see more viruses. In plants, the transmission of viruses is a little bit different than directly through infected individuals. So the viruses that we often get from coughs or sneezes from other people. Plants have pretty good defenses. They have barriers that keep viruses out. But if those barriers are harmed, like via cutting of a plant leaf or stem, viruses can be transmitted by mechanical means, things like mowing. And probably the most common way that they're transmitted between plants is via insects. Insects often have specialized mouth parts that can pierce the leaf or the stem of a plant, extract sap, and then the saliva can transfer viral particles. It's also possible for viruses to be transmitted via seed or pollen in some cases, but not for all viruses. So on this slide, you can see some symptoms that are common to viruses. In the middle, there's mottling. That's kind of the yellow patchiness on rose leaves. It's very common for viruses. On the right, you can see some wheat plants that have kind of streaky tips. It's another common symptom of viruses. And on the left, those are viral particles observable with an electron microscope. So other common symptoms are stunted height, twisting, and crinkling of the leaves. So as we're developing crops, we want to be particularly cognizant to make sure that we're maintaining high diversity that will allow the plants to resist infection, so genetic diversity, and also that we have methods and management of ways to control disease. In perennial plants, it's a little bit different system than with annuals, and there's a possibility that viruses or other pathogens could accumulate over time because there's living tissue that's present for many years. Sorry, I'm getting a ton of chats. Let me just look real quick. Okay. Sorry, that was a little bit distracting, but okay, come back to the presentation. So in perennial crops, we have living tissue that's there over multiple years, and the possibility that pathogens can replicate and increase in infection over multiple years, versus in an annual system, like on the bottom, where you have a different plant species every season that might have its own pathogens, but when the plant species is replaced, we get to kind of wipe the slate clean and start over again the next year. And there's also some other management practices that we use with perennials, like mowing and swathing and haying, that may transport or transmit viruses between plants more than in other systems. So we're particularly interested in making sure we don't have high infections of viral pathogens in perennial plants. Go on. So I'm going to talk about one virus in Silphium. And Silphium is a native member of the sunflower family that David Van Tassel's group here at the Land Institute is working on domesticating. They're using it as a perennial oilseed crop, and the oil could be used for cooking. The seed can be ground into meal, and they've even experimented with making things like tofu. So it's a very drought tolerant crop, very deep rooted, and provides a lot of floral resources also for pollinators. But we started noticing a couple of years ago that we found these symptoms in the field where the leaf veins were clear and the leaves were very twisted and kind of constricted at the tips. And we've been kind of trying to figure out what's causing this ever since. This can be pretty severe. In some fields, every plant in the field shows these symptoms and doesn't produce flowers. They can also be very stunted or about half the height that the plants usually are. And we noticed really severe symptoms, especially when the fields are waterlogged. And so initially we thought it might be caused by a nutrient deficiency or drought or flooding. But we did some nutrient testing and didn't find anything to be deficient. And the pattern that we saw, these symptoms were not really consistent with drought or flooding. And so over the summer, David was doing some research and he came across a virus of lettuce, a big vein leaf virus of lettuce. And the symptoms were really similar to what we saw in Silphium. And so we found a test for that virus, and we tested our Silphium samples, but they came back negative. And we also tested about 19 other viruses that are common in lettuce and sunflower, as well as viruses that are just common across a broad range of hosts. But all of those were also negative. And so during the fall, I guess about October, we collected some infected plant tissue and sent it to a company in the Netherlands for RNA sequencing. And so they took the plant tissue, extracted RNA from it, and got about 95 million mapped reads. The average coverage depth was 147 times, so that's the total number of copies of each of the sequences that we got, which is just a metric that describes kind of how well the sequences are covered and how much error there might be. But that was a very good coverage depth. So based on the sequence alignment, so they took all of the sequences that we got from the Silphium and aligned those against all known viruses, and found the very best candidate, where about 90% of the sequences aligned, was Dahlia mosaic virus. And you can see the coverage of the genome and the horizontal dashed lines that go across. So those are the sequences that we have. We represented a little over half of the genome with the amount of sequencing that we did. And then the vertical colored lines, that's the coverage, or the individual sequences that align on top of each other. So we started looking into Dahlia mosaic and found that it has 3 different strains. The most common is DMV-D10, And that's found in quite a number of host plants, dahlia, but also other members of the composite or aster family. And that particular strain of the virus is kind of interesting. It incorporates its DNA into the plant's DNA. And that means that it's also transmitted through the seed of the plant. So it can be kind of hard to get rid of it, or it can be present in a lot of the individuals of a population. And then there are two other strains. There's a dahlia common mosaic virus, and then there's just regular dahlia mosaic virus. Both of those are transmitted by aphids. So we started looking at the symptoms just to compare what we'd seen in Silphium with the symptoms that are found in dahlia. And they look kind of similar, at least in terms of the leaves and the vein clearing. The dahlia was also described to have some mottling, which we haven't seen in Silphium, but viral symptoms can be highly variable. And a lot of times plants that showing no symptoms at all can also be infected. One thing that was kind of interesting about this particular type of virus, it's called an endogenous pararetrovirus, is that the other viruses in this group can be induced by abiotic stress, things like drought or flooding. And so we're kind of wondering if what we've seen with entire fields displaying symptoms is because those plants have, or the field has high clay content and often is waterlogged, where in other fields we see much patchier display of symptoms. So right now we're currently trying to figure out which of the three strains of dahlia mosaic virus we have. And we have a colleague at Washington State University named Hanu Papu, who's been very gracious and has worked with us. He's studied dahlia mosaic for a number of years and was willing to test some of our Silphium samples. And so both at Washington State University and at the Land Institute, we've been using PCR-based approach, a polymerase chain reaction, to take the DNA extracted from the plant tissue and depicted in the tube here, with the gray DNA being the plant DNA and the red DNA being the viral DNA. And what we're doing is using a small sequence from dahlia mosaic virus that's known and mixing it in with the plant DNA that we think has the virus, and using amplification with an enzyme to increase the viral DNA. So in the bottom tube, you can see more red viral DNA, and then we can look at that viral DNA on a gel based on an expected size of the viral fragment. So that's actually kind of similar to how the COVID-19 test was working, when it kind of the slower version that was taking at least a day to process samples. And we're kind of at the point where we don't have a test that's working quite yet. So this is kind of like in the early days of COVID-19. Actually, the test designed for dahlia has shown up that we're getting all negative results, both at the Land Institute and Washington State University. And so now what we're doing is going back to the sequence that we generated from Silphium that's infected with the virus, and pulling out some of those viral sequences, designing new sequence probes, and trying the PCR with a number of different viral sequences to see if we can amplify part of that in the Silphium samples and have a way to test all of the plants in the breeding program and plants in the wild. So we have a lot of questions still to answer, and not nearly as many people working on this as are working on COVID. But some of our main questions right now are first, what strain do we have? Is this virus seed transmitted, or is it insect vectored? That's relevant for the management. And how does it spread? And then we're also wondering how common it is in our breeding program, because if it's in 98 to 100% of our plants, we probably are going to need to figure out ways to either eradicate it or find new germplasm that doesn't have it, or find plants that may have the virus but still can yield well and are kind of tolerant to the virus. And then we're also wondering what conditions result in the expression of the symptoms and presumably the viral replication also, and if we can avoid those types of conditions. So maybe it's avoiding really heavy clay soils or something like that. So that's kind of where we are, and I'd be happy to open it up for discussion now.
Great, thank you, Kathryn. We did have some questions come in. Please feel free to keep messaging myself. We can touch on some of the things Kathryn spoke about. So one of the questions, Kathryn, was how widespread is DMV?
So it's known worldwide in Dahlia populations. There have been studies done in Egypt and China, across Asia, and then throughout the United States. I think in every state, they've researched or looked at nursery stocks and have found cases of DMV in all the states. And then there's also like some other species that are kind of surprising, where they found it like in peppers, hot peppers in Asia, and then in a number of the plant species in the aster family. There was a study done in Kansas of a lot of I'm trying to remember. I think it was about, there were 5 different botanical gardens, and they sampled about 40 different species and found it in about a third of the aster species. So it's, yeah, fairly common.
Great, thank you. Another question somebody's asking is, do you only work with Silphium at the Land Institute, or are there any other research crops that you work with?
Yeah, so I work quite a bit with Kernza, intermediate wheatgrass. Kernza is the trademark of the grain of the plant, intermediate wheatgrass. There's a fungal disease that causes head blight and produces toxin in the grain. So that's a large component of what I work with also. And then worked a little bit with legumes, with alfalfa, and just trying to identify what other diseases are present in perennial sorghum and perennial wheat as well. So it's pretty diverse.
And what are some of the recent discoveries or exciting kind of highlights you've had within the programs lately?
So there's another pathogen of Silphium. It's a rust of a fungus that causes kind of a rusty lesion on the leaves and reduces the seed size and the yield of the plants and the photosynthetic capacity. But for that fungus, we've been trying to culture it and to store it in the lab, so that we could select individual plants that are resistant to the fungus, because we see quite a number of them in the field that are resistant, but the conditions in the field are kind of variable. In some years we get very little rust, and so that's kind of a problem for breeding, because we don't know which plants to select and which ones are really resistant. But we want to be able to do those type of trials in an inoculated setting in the field, or even better in the greenhouse. So we've been working on trying to store the spores of that fungus, and we started out over the summer and could store them for about a week or 2, and now we're up to 3 months, so and counting, but we're excited about that. And then, yeah, there are a few different things going on with that particular fungus.
Great. Are there any other plants that are affected by DMV?
Yeah, so besides dahlia, there are probably, I mean, quite a number of plants in the sunflower family and the aster family, including hot peppers, but I don't think those infections are very common. And kind of the early reports indicated that natural infection was only present in dahlia. That was research coming out of the 70s or done in the 70s. And that only in lab situations or with really high amounts of inoculum could other species be detected. But I think more recently that's been, we've found that there is natural infection in other species too.
Great. What are some of the 5-year goals you're looking at for the CPG program as a whole?
Oh, that's a good one. Yeah, having methodology that we could test large groups of plants from each of the breeding programs would be is something that I think is achievable in 5 years, at least for pathogens that we kind of have already identified at this point as being significant and needing to improve the genetic basis of those traits. So having a really fast, high throughput testing methodology. This is sounding a lot like COVID, but yeah, but it'll take us a little while to get that developed.
And why, as a plant breeder, did you want to focus on diseases?
So diseases end up being very important to yield, which is important to the bottom line and making the crops profitable and just viable as or economically viable to be planted. So to get any of these crops out on the landscape, we need to probably increase their yield or their profitability to some extent. And pathogens are yeah, it can be very devastating. They can completely eliminate the grain yield or probably in minor cases, at least reduce it by 10 or 20%.
Great. Well, that looks like all the questions we received. If any questions come up after the fact, or if you have any questions about TLI generally, please feel free to reach out. My personal email is my last name, which is Schneider, SCHNEIDER at landinstitute.org. I'd be happy to answer any questions or direct you to the best person to answer those. In the meantime, we hope you head to our website and stay in tune for some of the upcoming series we have. Next week, we have Dr. Ebony Murrell doing the Lunch and Learn. That'll be on Wednesday, April 15th. And we have some other exciting technician takeovers and perennial practice videos coming up soon. So we hope you can follow along. And if you'd like to stay up to date on kind of some of the latest updates and highlights coming out of the Land Institute and ways to engage and opportunities to help push the work forward, please visit landinstitute.org. But other than that, we hope you have a great rest of the week. And one last time, thanks, Kathryn.
All right. Thanks, Will.
Yep. Thank you all and have a great day.